This is a scanning EM. It's a fruit fly egg or a Drosophila egg almost immediately after fertilization. It's still a single cell. It's about maybe half a millimeter or 500 microns long. You look at the surface, you don't see anything very much. If you were to be able to look inside, you'd see that in this zone here, there's the nucleus of the single cell, which is the product of the fusion of the female pronucleus and the male pronucleus. You look at it, and it doesn't really look very interesting. The amazing thing, though, is that what this embryo has to do to become interesting is to convert from being a single cell into a multicellular organism where individual cells can assume different fates and begin to do different things. Now, one of the interesting things about embryonic development in Drosophila is that the embryo is able to do this extraordinarily rapidly, such that after 2.5 hours, this single cell has now transformed itself into a multicellular organism with now 6,000 cells, about 100 cells along the anterior, with the future head, future tail, anterior, posterior axis of the embryo. All the cells look pretty much the same. If I'd fixed this embryo for scanning M about 5 minutes later, what you would have seen is that now all of the cells in the embryo are no longer the same shape. There are clearly distinct things happening. There's an area here that's ultimately going to form the head of the embryo that's marked off by this fold that's called the cephalic or head fold from the rest of the embryo. There are other things beginning to happen in the embryo. Clearly, by this stage, at this point, 5-10 minutes later, the cells are marking they're showing distinct behaviors and showing how different they are. We can actually watch these behaviors in living embryos by tagging the surfaces of cells with fusion proteins between GFP and various membrane proteins, so we can follow individual cell shapes. And we watch this movie again, which you can see. This is again the blastoderm stage that we talked about. And watch in this area here, all the cells are pretty much the same, but right about there, you can begin to see this fold happening, and then you begin to see remarkable changes in the behaviors of cells. You can see this movement of cells as they sweep and move around move around the end of the embryo. Obviously, as you watch the process, and we'll watch it one more time, all the cells are the same. Individual cells begin to become distinct and with extraordinarily reproducible patterns. So embryos always make a head. They always make a head right here. They're always separated by folds. This behavior here, these cells here that are moving into this imagination here, are ultimately going to form the endodermal or gut regions of the embryo. We could roll this embryo over, and now we can watch from the ventral side, and we see that other things are going on. You can actually see a little bit of this head fold here, but what's more striking in this movements of cells. Again, watch right here. You'll see a fold forming. These are future muscle cells that are going to be brought into the interior of the embryo, because obviously that's where the embryo wants to have muscles.